Alright, in this video we're going to talk very briefly about proteins and how they work, how they fold actually is one of the main topics here. We want to understand how you get the shape of the protein. So we know from one of the previous videos that proteins are made from chemicals called amino acids. All right, and there are in humans anyhow, it's a little different. Not every species uses exactly the same. Some have a couple of extra amino acids that they might utilize. But in humans anyhow, we have 20 main amino acids. There is one other one we sometimes use called selenocysteine, but generally it's 20 to choose from. Oops. So these 20 are just like the DNA, where the DNA you had 4 letters. Here you have 20 letters to choose from. Now, I prefer to use 3-letter descriptions instead of one. Like there's an amino acid called proline. Well, proline can be represented as a P, or it can be represented as PRO. So I usually use PRO to represent them, to represent the amino acids. And here, I have an image, a kind of a chart of the amino acids here. Let's see if we can get that at full screen. It's a little big. All right. So I'm not going to make you learn all of the amino acids, and I'm not going to make you learn the structures either. But I do want to kind of use this chart to kind of show you that they are all chemically a little different, and they have different properties. These are positively charged amino acids. These are negatively charged amino acids. These have uncharged, what are called polar side chains, and these are special cases that have special abilities, you might say. And then here are the ones that are hydrophobic. And if you remember from maybe Bio 1 or from our talk about lipids, hydrophobic means it's afraid of water. So these are not going to like to be on the outside of a protein. These are going to be much happier chemically if they're on the inside. All right, so if these are on the inside, they want to be surrounded by the others. This is one of the reasons proteins fold, but we'll talk about many reasons, and that's what I want to get at here. So let's see here. Let's just go ahead and close this. I just wanted you to see that, so here's proline P, glycine is G, selenocysteine, which is actually a special case, is U, C, Q, N, T. These are what they look like, but I especially want you to realize that some are positive and some are negative. So what happens inside your body is, inside your cells, are that the ribosome starts assembling a protein. So ribosomes assemble the proteins out of amino acids. So the parts are the amino acids, and when they're done, the amino acids make a long string, kind of just like DNA. So, you might get P, G, A, let's see, L, here's another P, a C, a G, A, L, L. Okay, so that's how, in DNA we just had A, C, G, T. Here we have 20 letters to choose from. So you're going to get a long chain, just like you did with the DNA, but it's single-stranded. It's not going to be double-stranded. It's just a long chain of these chemicals. And so over here I'm going to represent these little chemicals as circles. And we're not going to bother with giving them names. I've shown you what they look like. But in general, the ribosome is going to take all of these and attach them. They're going to bind these together according to the instructions from a gene, which we will learn about. Hopefully you learned a little bit about that in Bio 1. Now, as this ribosome is creating these, they're going to come off the ribosome one at a time, but these amino acids have properties that I just showed you. Like maybe this one has a positive charge, and this one has a positive charge, but this one has a negative, and let's say this one has a positive. Well, the little bonds in between here are flexible. Not super flexible, kind of like our hairpin loop that we looked at, where we looked at that and we realized the DNA could kind of bend. Well, so can these. These can bend. Now, what also do we know? We also know, if you ever took physics or chemistry in high school or even in college, that what? The opposites attract, and if it's the same charge, they repel. So these two are going to repel. These two are repel, but they're right next to each other, so there's not much they can do about it. But they're too close for this to bend over and touch this one. You know, these little lines here, these bonds, can only bend so much. This one would be attracted to it, but this isn't going to be able to bend over and touch it. But this negative down here and this positive down here, they might be able to just bend enough so that this one can bend over and be attracted and fold into that one. And that's exactly what we see with this type of protein. So here maybe we have a negative, and here we have a positive, and this thing might fold over. All right, here, let's put a few more on here just to kind of get the idea. So that means I would put another one here and another one here, or a couple more here. All right, so here is our negative, here is our positive, and we have what, 1, 2, 3, 4 in between. All right, so what I'm saying is that when this is made, just like the DNA kind of moves around, it's a little flexible like a flag in the wind, and so it moves around for a while and it can fold over, and the positive and negatives will attract one another, and therefore you will end up with a folded protein. So whenever I draw a protein, I kind of draw it like this. All right, just all kinds all over the place. Well, that's to indicate that it's folded. If it's a long straight line, it's not folded. And the folding gives it its shape, its three-dimensional shape. And the three-dimensional shape is one of the main ideas of how proteins work. Not only do the charges here affect the chemistry of the protein, but the shape of it affects the chemistry of the protein as well. So the chemical nature of the outside, the chemical nature is decided by each one of these. Each one of these amino acids has a slightly different chemical ability, and you put them next to each other, it's going to affect that chemistry. So the exact nature of a protein's abilities are dependent on what the sequence of amino acids are, and then not only that, but it also controls the shape of it. So the shape of it allows it to like point something in a particular place. For instance, maybe you want this particular amino acid because it has some chemical property, you want it sticking out of the rest of the protein. Well, that might do it. If the rest of the protein curls up like this, and this one curls up over here like this, and this piece sticking out, that's what you want. That's the part that's reactive to do what you want. Well, the shape is important. There are proteins that act like scissors. Well, of course, they're shaped kind of like this. All right, and they can move. This part in here will be very flexible. And you add some ATP, and man, this thing moves. It cuts just like scissors. There's something called a restriction enzyme, which cuts DNA. And sure enough, it looks like a pair of scissors like this. The DNA fits in here. It's a helical loop, and it just chomps down on it and cuts it into two pieces. So if this were your DNA, this thing would come along here and close down on it and cut your DNA right there. So realize that these proteins do all the chemical work inside your body, or I would say 99% of it, if not all of it, pretty close. And we saw what they do in the little video where you had the little thing that looked like it was walking. All right, there are so many things that proteins do. They do almost everything. And remember that they need ATP to change shape a lot of the time. Sometimes they change shape when there's a pH difference or a change in salinity or a temperature. That can also make a shape change. But most of the time, it requires your cells to provide them with ATP, and then they can change shape. But I wanted to give you this idea just to show you how proteins operate.